So, ladies and gentlemen, welcome to the Spectradyne webinar this morning that's being held by Dr. Jean-Luc Fréquin, who is the co-founder and CEO of Spectradyne. We're delighted to welcome Spectradyne today to discuss their new technology, their new particle analyzer. And today's presentation will be a deep dive that Dr. Fréquin will be giving on their technology and discuss its applications in the extracellular vesicles and several other spaces. We at Select Bio are delighted to host this webinar and are pleased to welcome you. In the interest of time, ladies and gentlemen, we'll get started. At the conclusion of the presentation section of the webinar, Dr. Fréquin will be taking questions. So you can either type in your questions in the Q&A box. Alternatively, you could raise your hand electronically at the end of the webinar presentation and we'll unmute you. And then you can ask your question in real time to Dr. Fréquin. So without any further ado, ladies and gentlemen, let's welcome Dr. Jean-Luc Fréquin, the CEO and co-founder of Spectradyne, to deliver the presentation. Welcome, Dr. Fréquin.
Hi, thanks, Anil, and welcome everybody. Thank you for joining and for your interest in our technology and hearing about our new products, the ARC. This will be a little bit different format than our launch event from last month. This is a brand new product, so it'll be a little more conversational and hopefully you can learn a little more details. So this will be a technology deep dive into Spectradyne's new ARC particle analyzer, which delivers single particle phenotyping, size, and concentration at the nanoscale in a single instrument. So let's get going. We'll start with a brief introduction about why we're here and the importance of particle analysis, and then get right into the details of the ARC. We'll talk about how it works, and I want to spend most of our time showing you measurement examples of various different kinds of particles in our key application areas. And then I look forward to a good Q&A session at the end. So we're all here because accurate particle analysis is a critical thing, right? To characterize the concentration and the size of particles is really important, largely because nanoparticle applications are growing rapidly. We all know about the COVID vaccines, which are nanoparticle-based, LNP-based vaccines. There are all sorts of new gene therapies being developed, and as well as nanoscale diagnostics that leverage the biomarker potential of extracellular vesicles, for example. Also, many fundamental biology applications where being able to accurately measure the concentration of the particles is important. The reason it's so important is because at this size scale, the size and concentration of the particles dictate very strongly their performance. So, for example, size dictates the diffusion rates very strongly at the size scale, and concentration is the dose of a drug if it's a therapeutic that you're working on. And so that's directly impacting the safety and efficacy of a product, or in the case of fundamental biology, controlling the concentration of particles in your experiments is really important for robustness. One additional feature now is that biological nanoparticles are produced in complex media, right? They're made in cell culture media, or they're maybe present in biofluids directly. So it's important that when we're analyzing these, measuring their concentration or size, that we be able to do that in a heterogeneous sample, and still make sure that we're measuring the right particles that we need. You're probably here because the technologies that are out there that you're trying to use to do this are limited. So I've grouped them here in terms of three classes of measurement methods. The first ones are diffusion-based methods, and that includes dynamic light scattering and nanoparticle tracking analysis, even the fancy ones with interferometric imaging. These technologies suffer when the particles get small because the particles are hard to image with light, right? They're smaller than the wavelength of light, and it's hard to measure them that way. NTA in particular well, DLS is an averaging technique, right? So it's not going to tell you the details of the distribution, and it doesn't tell you concentration at all. But then NTA has the limitations of their limit of detection being variable depending on the sample. Particle immobilization techniques, this includes this interferometric imaging SP iris technique. They're really cool for phenotyping, right? But they don't deliver concentration measurements. They do count particles, but it's just a relative amount between different spots on the immobilization surface. And then there are single particle flow based technologies like dedicated nanoscale flow cytometers, or just regular flow cytometers that have been trying to push the limits to smaller sizes now. These ones have similar limitations because we're still trying to use light to detect particles that are smaller than the wavelength of light. And for flow cytometry particularly, in order to obtain size information from scatter, you have to be able to understand the optical properties of the particle very carefully or very well. And that's a challenge, especially in these heterogeneous samples where all these particles live. You'll notice too that every one of these techniques is an optical method, right? And so that gives a certain perspective. It's important to have different perspectives though. So they have all sort of similar limitations. They're indirect measurements of the size because we're looking at the scatter. And in many cases, those indirect measurements lead to misleading results. And that can slow down your science, and that's not good for anybody. So there's definitely a critical need still in this context for accurate nanoparticle size, concentration, and phenotyping in complex samples. That's where Spectradyne's technology comes in. The ARC particle analyzer delivers single particle fluorescence-based phenotyping. Okay, and we're using quantitative, this is a quantitative fluorescence measurement, and we report results in mistraceable units. It has state-of-the-art sensitivity and detects up to 3 fluorescence channels simultaneously. This happens at the exact same time as we're using Spectradyne's proven technique called MRPS to measure the size and concentration of the particles. So we have accurate concentration size down to 50 nanometers, up to 2 microns on the ARC. These are direct measurements of particle size and concentration, and ultimately they deliver subpopulation analysis in complex samples, which is what you're after. Technology is easy to use. It takes about 5 minutes per measurement. And because we're leveraging all the power of microfluidics, it only takes a couple of microliters of your sample to do a measurement. The flow cytometry nature of the fluorescence measurements are compatible with all the infrastructure that's been developed for flow cytometry over the years, which is a very powerful toolkit that you can leverage on the ARC also. So let me spend a couple of minutes talking about explaining how the technology works in detail. The magic really happens inside this microfluidic cartridge. It's about a ½ inch on the side. The way the MRPS technology works is as follows. So we flow the sample through constriction in the fluid flow, okay, and we run an electrical current through here. And every time a single particle goes through this bottleneck, this sensing constriction, we get an electrical signal that tells us 2 things. One is the particle size, so that's how big the electrical blip that we get is related to the particle size. And secondly, it tells us the concentration of the particles because it tells us the flow rate of the sample through the constriction. So we're directly counting particles and measuring the volume in which they're counted, and that is the most direct measurement of concentration that you can get. Very straightforward. Now in the ARC, what we're doing is we're exciting the particles with a laser at the exact same time that we're measuring them with MRPS. And of course, if they're fluorescing, then they emit their fluorescence light at a different wavelength, and we detect that and quantify that. And so now in the ARC, we're doing size and concentration by MRPS and adding fluorescence parameters in up to 3 different channels simultaneously. So this is like flow cytometry plus our NCS1 to get accurate particle size and concentration in a single instrument. So I'll show you some examples of that. Obviously, this is a made-up plot, but in reality, it doesn't look that different. So this is a real data plot showing you MRPS measurement in blue. So this downward spike is the detection of a single particle by MRPS. And then the three simultaneous fluorescence measurements happening, at the exact same time. So we see a signal in red, we see a signal in orange, and a signal in green channels for these. This example is a polystyrene particle that is broadly fluorescent. So you excite it at 488 nanometers, and it emits across a broad band, and so we get signals in all three of our detectors this way. So the key impact areas for the ARC are these four areas. These will be our focus in launching this technology. Primarily in extracellular vesicles, where the ability to accurately measure the concentration and size and phenotype will lead to more robust science, because we'll be able to control your experiments better and enable new discoveries to happen faster in your labs, which is what we all want. In nanomedicine applications, we're talking about accurately measuring the concentration and the dose and the size distribution of specific drugs, even in a complex formulation or in a complex manufacturing environment. In virus and gene therapy applications, this is a really powerful impact, I think, because the ARC delivers virus concentration in about 5 minutes, specific virus concentration in about 5 minutes. Whereas if you've ever tried to do tissue culture-based analysis of virus concentration, it can take hours at best and potentially days, plus many, many tighter plates and things like that. So we're offering a huge time saving in the virus and gene therapy applications this way. Finally, and this is a little bit of a future project, is getting into nanoscale diagnostics, where biomarkers that are the nanoscale that haven't been able to be quantified properly in the past now can be, and we're working with partners to develop these biomarker applications separately. Okay, so let's get into some data examples. So the first example I'd like to show you is an analysis of a mixture of extracellular vesicles in which we're looking for specifically a subpopulation that are fluorescent. So first, we can look at just the MRPS signal. So we ran the sample on the ARC. And if we looked only at the electrical detection, so this is counting all the particles, and we know they're real particles because of the MRPS electrical signal that we get from them. We see this typical broad size distribution. So there's an approximate power law dependence of concentration on size, on particle diameter. But now, because we're on the ARC, we're not only measuring particle size and concentration, we're also simultaneously measuring their fluorescence, right? And so we can use that information to detect just the particles of interest in this sample. When we do that, what we see is a nice, well-defined population of EVs that are expressing these EVs express GFP on their surface, and they're showing up as positive fluorescent in our FITC channel. Now I'll get a little more specific about what it means to be considered FITC positive in a second when we talk about detection limits. So we can be quantitative over any size range here. So what we're seeing is over the peak of this population, we're measuring about 8% of all the particles are fluorescent. And we can measure that as a fraction, right, as a percentage of all the particles. So this is a single measurement showing concentration of the particles of interest, as well as their purity in a broader context of all the particles in the sample. It's in the single measurement. So we have ascertained that the fluorescent positive particles have a mean diameter of 110 nanometers. Their concentration, we can be quantitative, is about 1.3 times 10 to the 9 or 1.0 times 10 to the 9 particles per mil. And we know from their fluorescence that about 8% of the particles in this sample are GFP positive. Okay, so let's take an example from nanomedicine applications now and look at liposomes. So this sample that I'm showing you here is a mixture of 100 nanometer extruded liposomes that have PE on their surface, conjugated to their surface. And it's a mix of those liposomes with a spike in that we put in of 180 nanometer fluorescent polystyrene beads. And so in the MRPS size distribution, which shows all the particles in the sample, we can see, clearly two different populations. The liposomes are around 100 nanometers, a little bigger, and the 180 nanometers come in right here. We have a lot more information about this sample, though, coming. So when we look at now, this is a parametric scatter plot here now, where we're showing diameter measured by MRPS on the x-axis, right, and brightness, fluorescence brightness in the PE channel on the y-axis, and this is in absolute MESF units. So if you're not familiar with what these units are, I encourage you to go read about them. There's lots of articles about them, because these are what we need to be familiar with when we're talking about translatable data between labs. So if we're going to compare measurements, we need to be measuring the brightness of the particles in absolute units, and that means these MESF units, which are molecules of equivalent soluble fluorophore. So what that means is, if a particle is measured to have 100 units of PE MESF, that means it's as bright as 100 PE fluorophores floating in solution. That's essentially what that means. So when we have the excitation laser off, what we see is this scattered distribution. So most of the particles are all down here, right? And there's some events that are showing fluorescent positive here, but they're basically insignificant. This cluster here are the 180 nanometer YGs, and we know that because we're measuring the actual diameter of the particles, right, by MRPS. And these are the liposomes, right? They correspond to this size diameter. Now let's turn on the laser and see what happens. So when we turn on the excitation, the liposome population all of a sudden starts fluorescing in the PE channel, right? And so we see a higher MESF or a higher PE signal for these particles when we turn on the laser. The 180 YGs also show fluorescence. That's these populations here, this particle population here. Using this scatter plot, we can define a gate to gate purely on the liposomes, so we can exclude the 180 YG spike in that we put in. And we can collect only the particles that have a fluorescence above our detection limit in this case, which is 7 MESF units. So everything that's brighter than 7 PE molecules floating in solution, we're going to capture inside this gate. And then we're going to measure their concentration and size distribution. That's what we're showing here. So now the blue curve shows the same data we were just looking at, which is all the particles. And then the orange curve shows the liposomes that have a brightness of more than 7 MESF units in the PE channel. This is a cool example because this is a complex mixture, right? But we've shown a spike in of a known brightness of particle. I'll talk about that in a second next. And isolated using custom gating the population of interest and been now quantitative over whatever size range we want. But in the liposome region, we can see about 50% of the particles greater than 65 nanometers, brighter than 7 MESF units in PE are fluorescent positive. So this single measurement has shown these liposomes measure their size, their concentration, and their phenotype, as well as a quantitative measure of their brightness in terms of PE MESF. I didn't explain how we got this number yet, but I will in the next example. So as a third measurement example, I'd like to show a measurement of GFP expressing murine leukemia virus, or MLV. This is relevant for all virus quantification applications, including in gene therapy. And so again, we'll start with just the MRPS data. So this is showing the particle concentration versus size for all the particles in the sample as measured by MRPS. We know these are real particles and not artifacts of flow cytometry scatter measurements, for example, right? Because they're giving us a signal in MRPS. So there's a clear peak here from the leukemia, from the MLV already, but there's obviously superimposed on top of a background of other particles. So we can measure that concentration over the size range of interest here. It's about 3 × 10⁸ particles per mil. Using the fluorescence information that we have, though, we can count just the FITC positive. So these are expressing GFP. That's similar to the FITC fluorophore. And so in the FITC channel, these are considered positive events. And we can see that about 2/3 of them on the size range are expressing GFP on their surface properly. Now let's talk about being quantitative. So how bright are these? How bright are these particles? Well, so we can show the data in a different way, which is for all the GFP positive events here, we can show the concentration as a function of brightness in absolute units. So that's what this plot is showing here. There's a nice clear peak, obviously, in the brightness distribution, where we can quantify the concentration as a function of brightness for all the particles that are GFP positive. And from this, we can say that the average brightness of these GFP expressing EVs is about 760 FITC units. So each one on average is about as bright as 760 free FITC molecules floating in solution. So this is in a single 3-minute measurement. We've accurately measured the size of these virus particles, their concentration, their phenotype, and how bright they are. So how bright they are is relevant for maybe your modification of these particles, right? So maybe you're trying to get them to express a certain motif on their surface, or you're trying to quantify their cargo. These are the absolute brightness measurement is critical for assessing those parameters. So let's talk a little bit next about the sensitivity of the ARC in fluorescence detection, how we ascertain that, and what the limits of detection really are. So for this experiment, we'll use a set of cross-calibrated beads. These are polystyrene beads, the 180 YGs that we've been using already, and a larger size 290 nanometer diameter YG beads. These beads are broadly fluorescent, and so you can see that in this composite histogram here, or CST plot, you can see that almost all of the particles are showing up positive in both the FITC channel, which is the green distribution, and the PE channel. So they're expressing broadly, they're fluorescing over a broad range of wavelengths, and so they're showing up positive in both of these channels here. Okay, so how bright are they? Well, the standard practice in flow cytometry is to perform a cross calibration one time with NIST NESF standard particles, and so we send these particles out to an external collaborator. They perform this cross calibration. This is a one-time thing. So now we know that the 180 YG particles are equivalent to about 9,000 or 900 free floating molecules of FITC and about 20 free floating molecules of PE. And the 290 YG particles have the same, or have different numbers. They're brighter, right? Because they're bigger. So armed with these numbers now, we can say, well, so what's the signal to noise for these, and then estimate the limit of detection for our technology this way. You can also, as we've shown in many of these experiments, you can also now use these as spike-ins into your measurement for a built-in positive control for size, concentration, and absolute fluorescence brightness, right? So these are powerful tools for quantitative particle analysis. All right, so let's first look at the FITC channel, all right? So what I'm showing you here is a plot with concentration on the y-axis and absolute brightness in the FITC channel in terms of equivalent reference fluorophores. So these are not FITC fluorophores, but we're comparing it to how bright free-floating FITC molecules are with our setup. So obviously we see clearly two peaks. We've got the 180 YGs and the brighter 290 YGs. And then this is, noise in the detector, right? So this would be our noise floor. Now, one thing that's interesting is that if we align this plot to scale such that the 290s match the cross-calibrated value, right? This is using them as a built-in positive control for this measurement, the 180 YGs match their independent brightness assignment from the cross calibration. So we know the system is working, right? And now, given the separation between these particles and the noise floor, we can assess what the noise floor is here. Well, I'll do that after we look at the PE channel. So in the PE channel, we do the same thing. So again, we align the 290s to their assigned value. The 180s match their independent brightness assignment also. Now let's look at the noise floor. So for the FTSE channel, under these conditions, it looks like the noise floor starts kicking up around 20 ERF. So in the FTSE channel. So that means that we should be able to detect something that is as weakly fluorescing as 20 floating FTSE molecules in solution. In the PE channel, PE is a much brighter fluorophore, so our detection limit is of order 1 PE molecule floating in solution, right? So we're approaching the single fluorochrome detection limit here, which is super exciting. These detection limits are equal or better to many commercial flow cytometers on the market. Really proud of our development efforts on this one. Okay, so I think I've shown a few examples of measurements, as well as how we assign absolute brightness measurements to things, to our measurements. You can learn much more about the ARC on our website. We've got short videos, medium videos, as well as the full launch event available, recorded on our website. The launch event includes some of the examples I've shown today, some other ones, and an expert panel featuring Dr. Jones from the NCI and Dr. Edwin Vanderpool from UMC in Amsterdam. We also have much more information on the ARC product page on our website, so I encourage you to go and visit there. We have early adopter deals currently running for the ARC. So we're offering significant savings through September 30th or November 30th this year. Also, if you're an existing NCS1 user, we're offering special trade-in value for your NCS1 through the end of the next month. We're also offering NCS1 deals. So currently 20% off through the end of November. Please come take advantage of us if you want to learn more, contact our sales associates by email or visit our website. If you would like to see the ARC in person, there are a couple of great opportunities for doing that coming out soon. In November, we'll be in Waikiki. I can't believe it, but we'll be demonstrating the ARC in person there in the middle of November next month. That is an ICEV conference that specifically is showcasing technologies for quantifying EVs and also separation and prepping EVs. So we're looking forward to that, obviously. And then right here in Long Beach, that's where our office is. But this next Select Bio conference, extracellular vesicles, technologies, and biological investigations will be taking place right here in our backyard in December. We will have the ARC there also running live demos. So I encourage you to reach out to our sales team in advance so we can schedule one-to-one meetings so you can learn more. And then you'll be able to see this technology operating live. So thank you very much for joining us today. I hope you learned some stuff, and I'd be happy to answer any questions. I look forward to a good discussion at this point. Thank you.
Thanks very much, Jean-Luc, for a great talk as always. Ladies and gentlemen, as you can raise hands, and the first one is by Jonathan Burton. So Jordan, if you would kindly unmute and you could speak now and ask your question. Thank you.
Hi, Jean-Luc. It's been a while.
Hi, Jordan.
This is great technology. One thing I was wondering, you showed a lot of data where it was like, and you can correct me if I'm wrong, it was like FTSE or GFP like expressed in a particle. And I was wondering, like, for exosome work, potentially you would use an antibody to conjugate to the outer membrane markers of the vesicle.
Yep.
Do you have any data showing that, and also whether or not it shifts the size distribution of the vesicles when you have that conjugated antibody on it?
Good question. So yes, we have the data. And are you going to ICEV in Hawaii next month?
I don't think so.
Okay. There will be there's a really cool experiment that's going on where they sent out EV samples to all the vendors. We're participating in that, where the instrumentation vendors will do their best to measure those and be quantitative about the number of epitopes, CD81 in this case, on the surface of the EVs. So we have that kind of data. I don't have it prepared here for today, but we have that kind of data. We have other examples of specific antibody labeling. It's just as you would expect, like in flow cytometry. Your question about whether the tagging with antibodies affects the size is a really good one. And to first order, the answer is no. And the reason is because what we're doing, like you know, is we're measuring the size based on the volume of the particle. And so you would have to get a complete coating of antibody over the surface of the particle in order to change the volume appreciably. And so as far as we know, the data so far have not shown any indication that that's a significant enough change for us to detect a difference yet.
That's great. Thank you so much. Yes, it did.
Sure. Great. Hope to see you soon, Jordan.
Yeah, you too.
Thank you very much. Jordan, ladies and gentlemen, any further questions? Please kindly either type in the Q&A box or raise your hand and we'll unmute you. Whilst people are formulating their questions, I have one for Jean-Luc. So we see a number of companies in this space, Luminex and others, that have flow cytometers that are seeking to do EV analysis. Could you kind of give a space map of companies and how Spectradyne positions itself vis-a-vis these other companies to give the audience kind of like a guidance as to, okay, why should we be using Spectradyne versus Luminex or Beckman or other companies in the space?
Great. Yeah. Thanks for the opportunity to answer that one. So flow cytometry, all the big flow cytometry players are trying to push to smaller particles, right? And there's new products being released all the time that are attempting to rejig or modify flow cytometry to allow some kind of glimpse of what's at the smaller scale. Fundamentally, these technologies are all limited by the optical wavelength of light, right? So it's very hard to measure a particle that's smaller than the wavelength of light. And when you start trying to do that with scatter, you have to make all kinds of assumptions all of a sudden about the particle material, their index of refraction. And that's not easy. There's, people, they're doing a lot of great workarounds for trying to calculate that and build models for how to translate scatter into size. But that's complicated. And as soon as you have a heterogeneous sample, like most real samples, then all that machinery starts breaking down, right? It's hard to do that for when you don't know a priori what the particles are made of or what their index contrast is. So all that to say that the ARC, we position the ARC as strongly complementary to the flow cytometry technology. So, flow cytometry has a fundamental limit for how small particles can be detected in there. We are offering a completely orthogonal method for measuring the size and concentration, but still benefiting from the fluorescence technology that, that's so well developed and so powerful, right? Fluorescent labeling of particles is a super powerful technique. So we have integrated those technologies into a single box, right? And you could think of it as extending the size range of a flow cytometer down, but it does really provide completely new information that way. Thanks for the question.
Thank you. Any further questions, ladies and gentlemen? The recording of Jean-Luc's presentation will be sent out to all the attendees in a few days' time, as well as, I believe, on the Spectradyne website. But this is a good time to ask any specific questions that Jean-Luc can address for you.
I just wanted to jump in to add to what Jean-Luc just said. I was busy, so I might be repeating him, but one of the things that's really neat about this system is that you are getting two truly orthogonal measurement techniques in a single system. those of you familiar with the MySev initiative know that they have recommended for EVs in particular that you should be using orthogonal techniques. And I think that's true of any particles that you measure, but in the EV space, MySev specifically calls that out.
Yeah, I think that's a great point. The fact that you have multiple independent methodologies to study EVs and to provide that as proof is important. There's a question that came in the Q&A box. It's by Sandra Prassel. I think that's a German last name with the German double S. So I'll read it out. Is it possible to do compensation in the software for two fluorophores which show considerable spillover like FITC and PE?
Yeah, great question. So that's under development for right now, but it won't be long. The software is moving fast. So that's something we plan to offer in the next few months. So it'll be ready for you.
Great, thank you. Any further questions, ladies and gentlemen? If not,
So,
yeah, go ahead, please.
Sorry, I'll just add to that we're also developing an export tool that will allow conversion of the data to standard flow cytometry formats like FCS format. So if you are familiar with using standard flow cytometry tools like FlowJo, then our data will go straight into there and you'll be able to use it in your conventional ways.
Thank you. That was very important. Any further questions, ladies and gentlemen? If you have, this is a good time to ask. If not, we'll ask maybe Jean-Luc as well as Lou to give some final closing thoughts about, a few bullet points about the technology and key takeaways from the webinar. So both Jean-Luc first and then Lou, please. Thank you.
Sure. Yeah. So, I mean, I hope you've learned about how the ARC works and seen some impressive measurements today. And ultimately, we want to hear from you. So if there are certain application examples you'd like to see, or if you would like to send us samples to measure on the ARC, we'd be happy to do that and work with you to see how well it can help your research. So I encourage you to reach out to our sales team for that. And you can do that either through this email address at the bottom or by visiting our website and sending an inquiry that way. So looking forward to hearing from you. Thanks very much again for your time today.
Yeah, I don't really have much to add to that, other than to re-emphasize what Jean-Luc said about sending us samples. This is new technology, and we are learning some amazing stuff as we get samples from the field. It's really an enabling technology. I don't think we've scratched the surface yet of the potential for this product. So we're really encouraging people that have specific applications in mind to send us some samples so we can learn along with you the potential uses.
Thank you to Lou and Jean-Luc. I have one follow-up question. Can people do custom services with Spectradyne? In other words, send 10 samples and have the work be done on site at your Long Beach facility and get the data, as opposed to, as a way to test out the system on a fee-for-service model?
That's a good question, thanks. Ultimately, we'd like to get the instrument in your lab, but we want to do what it takes to get to demonstrate its power to you, right? And so, if you have ideas, we want to hear from you, and we can discuss all kind of arrangements. To first order, we're happy to measure a few samples for free just to show you how cool it is. So just reach out to us and let's try it out.
Yeah, on that note, we'd like to thank Jean-Luc as well as the Spectradyne team for the webinar today. And they'll be sending the link to the recording, which will also reside on their website. We'd like to thank you all for participating today, and we look forward to welcoming you to the next Spectradyne webinar. So thank you, everybody, and hope everybody has a great day.
Thank you very much.
Thank you.
Bye.